Good afternoon to all the lecturers and my peers. My name is Goh Mei Rong and I'm going to propose my FYP project under the supervision of Dr. Tan Yi Sin. First of all, please take a look at the white fluffy stuff on my slide. This is the oyster mushroom that everybody loves to eat. But what if I tell you that they are not edible because they have been contaminated with the green mold disease? In order to cure the mushroom, we will first have to find out the different trichoderma strains that trigger the green mold disease using a molecular marker known as inter-simple sequence repeat or ISSR. Oyster mushroom is scientifically known as Spiritus species. It is a macrofungi belonging to the class Basidiomycetes and family Agaricaceae. Oyster mushroom is named for its fan-shaped caps and grayish-brown color which resembles the shell of an oyster. It contains all the essential nutrients that increase its food and medicinal value. Oyster mushroom is one of the few carnivorous species that can eliminate bacteria and nematodes in the agriculture. Living in a hot temperate country like Malaysia, we have the opportunity to cultivate the various types of oyster mushroom. They are saprotropic and resilient enough to be cultivated on the agro waste, such as rice straw, sawdust, and cotton. However, the crop of oyster mushroom around the world has been severely affected by the green mold pandemic. This pandemic is not due to the virus, but a macrofungi known as Trichoderma species. The strains of Trichoderma can be varied among the mushroom farms in different countries. The Trichoderma species initiate microparasitic activity with hydrolytic enzyme that lies the mushroom cell wall. Besides, mycotoxins from Trichoderma species will also retard the mycelium growth. The trichoderma will only turn green upon heavy sporulation, which leads to the death of mushroom under exhausted nutrient and space. Throughout the years, 3 markers have been figured out to classify the biodiversity of Trichoderma species. In 1969, scientists had divided genus Trichoderma into 9 aggregates based on the conidiophore branching and conidia shape. However, the refined classification does not accommodate the full range of morphological variation and later been revised by BISAIDS into 5 sectional levels. With the advancement of analytical tools, high-performance liquid chromatography could be used to generate chromatographic image for the fungal extract. The fungal strain can be identified by looking at the peaks or background signals on the chromatograms. Nonetheless, the analysis can be unstable due to environmental impacts on the secondary metabolites. The first genetic marker used in fungal systematic is the RFLP, but it is soon replaced by other molecular markers due to tedious process. In 1990, RAPT developed by William was able to perform well with just a few nanograms of DNA. However, it is a dominant marker which cannot distinguish between homozygous and heterozygous individuals. Nowadays, most of the studies are using the ITS marker, which is considered as the universal barcode for fungal systematics. On top of these markers, my project will focus on using the ISSR marker. The genome of every eukaryote contains the ISSR region, which is the tandem repeats of 1 to 6 base pair. The region flanked by SSR is known as ISSR. This ISSR region will then undergo amplification with primers produced from 3' or 5' anchor SSR sequences. Each individual has a unique number of SSR sites, and thereby showing different number of bands on agarose gel. In this context, we hereby hypothesize that the ISSR can provide molecular differentiation between the Trichoderma species. The objective for this study is to review the classification techniques and investigate the genetic diversity of Trichoderma species using ISSR markers. My project will first begin with a literature review. After that, the 20 Trichoderma species will be collected and cultured in PDA. The fungal DNA will then be extracted using the InuPrep plant DNA kit. The purified fungal DNA will later undergo PCR amplification with 6 different ISSR primers before taking the PCR amplicons for electrophoresis. Finally, the band scoring of DNA fragment will be conducted. At the end of this project, we expect that the use of ISSR marker can yield the genetic profile of different Trichoderma species, which help us in better identifying the causative agent for local green mold disease. This is the Gantt chart and references. Thank you.